So what I've started doing is kind of making an animation of some of the different ways that cell signaling can occur. It's definitely not complete by any means, but I want to take a second and go through some of what I've done so far with you. So on the kind of brown line here, you can see that's going to be the cell membrane, and there's a lot of stuff going on. For starters, we've got a G protein coupled receptor, and so there's the one end of the amino acid of the protein, the polypeptide, is kind of on the outside, and it kind of goes back and forth through a couple of times here, and there's going to be a binding site right here for the G protein to actually attach to. Here's the binding site for the ligand, so we can go ahead and hit play and see what's going to happen. So what you should be thinking about is the ligand binding, changing of conformation at the inside of the protein, the intracellular area, attachment of the G protein. We'll see here that GDP is then going to be kicked off. GTP can then bind. When the GTP binds to the G protein, that's going to activate adenylyl cyclase, which can then start converting ATP into cyclic AMP. Also on the membrane here, we'll be going through receptor tyrosine kinases and ion-gated channels over here. I haven't had a chance quite yet to animate the phospholipase C and go through everything going on with IP3, PIP2, IP3, DAG. That's what I meant to say, and therefore calcium. But hopefully this will give you a little bit of an understanding of some of what's going on. So here we go. If we hit play here, we can see the ligand binding. That's going to change the conformation of the G protein coupled receptor so that the G protein can bind. You can already see right here that GDP was kicked out, and that's going to allow GTP to bind, activation of adenylyl cyclase, so that then ATP binds to cyclic AMP. Also, I want you to take a notice here that the GTP that was bound to the G protein was cleaved off and reconverted into GDP. So one of the phosphates would have been cleaved. Okay, I'll go back through all of this and play it again without me explaining it. But anyway, next we're going to take a look at the receptor tyrosine kinases. Remember with these guys that we're going to need not one but two ligands binding. That's going to cause dimerization and cross-phosphorylation. So this guy is phosphorylating this side, this guy is phosphorylating this side, and now the receptor tyrosine kinase is active and can then activate other downstream products. Okay, and finally we've got over here the ion-gated channel. That's going to open up the channel for you. When the ligand binds, the ligand leaves, and then the channel is back to being closed. And this is passive transport, so we should be thinking about things moving from an area of higher to lower concentration. Okay, let's go ahead and just play the whole thing kind of back to back. Really big picture here, okay? We're looking at three types of reception: a G protein coupled receptor, a receptor tyrosine kinase, and an ion channel receptor. One of the kind of downstream, that transduction cascade, I've shown adenylyl cyclase making cyclic AMP. There we go. So that would be part of the transduction cascade, as would the activation of phospholipase C, that I don't have shown yet, but we'll get there, right? Okay, anyway, let's go ahead, restart, and show a little bit of everything that is going on. Start from the beginning, zoom in here, boop, boop. Okay, sorry about that. Try that again. All right, as a reminder, three different cascades would be shown here. So the ligand that came over here and bound to the GPCR would be a different pathway than that of the RTK, which would be different than the ion channel here. But I just wanted to show it in one quick animation for you. So if you've got questions, as always, get in touch with me and let me know, and hopefully this helped a little bit to elucidate a pretty complex topic.